Good morning, everyone, and welcome to the inaugural Broadacre Cropping Initiative Research Showcase. I'm delighted you can join us here today at USQ Toowoomba campus and online. I can't actually see how many people are online at the moment, so hopefully there are lots of people. My name is Professor John Bell. I'm the Deputy Vice-Chancellor Research and Innovation here at the University of Southern Queensland. I'd like to acknowledge the traditional custodians of the lands and waterways where the university is located and where all of our online guests are located also. We celebrate the continuous living cultures of First Australians and acknowledge the important contributions Aboriginal and Torres Strait Islander people have and continue to make in Australian society. The university respects and acknowledges our Aboriginal and Torres Strait Islander students, staff, elders and visitors who come from many nations. So before continuing, I have some important housekeeping for today's events. You can probably guess it's about COVID mostly. This is a COVID-safe event. Our event today is hosted at a venue with sufficient capacity to accommodate everyone within the guidelines currently enforced for COVID-19 in Queensland. Please ensure you've registered via the QR code. You'll see signage around the registration desk for those who haven't. Restrooms are out the doors, out the second set of doors and to the right, excuse me, outside the building. In an emergency, please exit the building via the nearest safe emergency exit. So there are emergency exits in 3 locations here, and follow any instructions from USQ security and building wardens. Should you have any questions throughout the day, please talk to one of the very efficient and effective USQ team, Liz, Kirsten, Will and Juanita. So I'm the MC for this morning's official proceedings. I'd like to, first of all, acknowledge our distinguished guests joining us today, Professor Rdne Mackenzie, the Vice-Chancellor of the University of Southern Queensland, Bob G, who's online, the Director General of the Queensland Department of Agriculture and Fisheries, Bernadette Ditchfield, Deputy Director General of the Queensland Department of Agriculture and Fisheries, who's here today on campus, Julia Playford, Executive Director from the Queensland Department of Environmental Sciences, or Environment and Sciences, I think, yes. Professor Gavin Ash, Executive Director of the Institute for Life Sciences and Environment at USQ, and Chris Downs, the General Manager for Crop and Food. So the Broadacre Cropping Initiative means KISS, so a very interestingly named initiative. The Broadacre Cropping Initiative is a partnership between the University of Southern Queensland and the Queensland Department of Agriculture and Fisheries. This started in 2014, and the partnership's proven to be very efficient and effective research alliance that's taken advantage of the strong synergies between USQ, the research that we do here, and our friends at the Queensland Department of Agriculture and Fisheries. We are very proud of this important and highly regarded partnership. So to start the official proceedings for the day, I'd like to introduce the USQ Vice-Chancellor, Professor Rdne Mackenzie, to say a few words.
Thanks very much, John, and it's my absolute pleasure to welcome you all here to this wonderful event on behalf of the university, also acknowledging those online, and I know Bob G's online and I had the pleasure of meeting with him very recently at a function. I'd also like to acknowledge the traditional custodians of the land on which we're all meeting today, and also to acknowledge the significance of what those many, many tens of thousands of years are to what we do today in caring for the land in particular. It's really exciting to have you all here, and I know that we've been very much looking forward to it, and the Broadacre Cropping Initiative has been a wonderful collaboration. Our flagship areas are in space and defence, agriculture, health and regional development, and as you can imagine, they're all interconnected. As we have launched our new strategic plan, 21 to 25, these things are our absolute focus moving forwards. And why they're our focus? Because they are of vital interest to the communities that we serve. Industry links, of course, are vital to everything. And a three-way collaboration like this, where we have the Queensland Department of Agriculture and Fisheries, where we have the University of Southern Queensland, and also industry coming together to solve problems that are really critical to this industry, is undoubtedly the way forward. We really pride ourselves on research that's regionally relevant, of national significance and global impact, and work in this area certainly is. So I'd like to just finish by wishing you the very best for today. I'm sure there's going to be some wonderful outcomes. I know that Gavin's been very much looking forward to it, and the rest of our researchers as well, and look forward to hearing how it goes. So welcome again on behalf of the university.
I'd now like to introduce Mr. Robert Gee, APM, Director General of the Queensland Department of Agriculture and Fisheries, and Bob is online, and I hope you can all see him.
Thanks very much, John. Can I too acknowledge First Nations people everywhere, wherever you are, online or in the room. I wish I was in the room, it just simply couldn't happen today. Can I acknowledge the support I've been given by the VC, Rdne and John in particular, already in my role. But today is about you and all those people online. Geraldine said it well. It's actually about a partnership between USQ, our department and industry. I'm a bit biased. I'm a graduate of USQ. I understand its strengths. If you stop and think where this started only 70 years ago, what a phenomenal success already. But for me, there is much more to do through the people in the room and online today. A great success already. I don't have to tell you what agriculture means to this state. 200 places across the world get our products. Despite COVID, ag's on the up still, particularly in grains and cotton. We'll see an increase hopefully this year. But for the work that you are doing, you are leading, I just wanted to give a plug for USQ. I think strong connections with industry, with rural communities, just shines through in everything USQ does. The vision I've seen through the VC, John, and the work and the research that's being done is outstanding, but it's all focused on strong outcomes for rural and regional Australia. Can I encourage everyone to keep up the work you're doing, to reach out and to question my department, my people, government, local, state and federal. Really reach out to us and question whether we're doing enough to support you. Our role, I think, is, and our commitment's clear. We'll be at 10 years in this partnership soon. I'm looking forward to it going for longer and longer. It's really exciting. Chris Downs is going to talk on behalf of DAF soon. He's a very valued member of our senior executive. But what's happening in terms of now this conflation and things coming together, this intersection of technology and engineering across industries is very, very exciting. And if we can keep supply chains strong, based on the work you're doing, protecting, getting in front of pathogens, whatever it is, the work you're doing is critical. Can I close there by saying that despite COVID, you're being successful. You'll get great support from us, recognise the initiative, but just also recognise the great potential it has to do more. And can I close by saying this, that COVID will be a test for Queensland as it moves through. We're well positioned. But for this group, the Department of Ag and Fisheries in Queensland, we're here 24/7 to help you. I can't help but say that to close, because it's really important. You can reach out to us to maintain business continuity. I really wish you well for the day. Thanks to USQ and industry. Thanks, John.
Thank you, Bob. We're very appreciative of your time and your comments when you joined us today. Thank you very much. I'd now like to introduce Professor Gavin Ash. As I mentioned before, Gavin is the Executive Director at the Institute for Life Sciences and the Environment. Over to you, Gavin.
Thanks very much, John. And welcome, everybody. Really happy to have you all here, to see so many people in the room, but also to greet the people online. And thanks very much for your kind words earlier, Bob. I'd like to thank the Vice-Chancellor and the DVC for making the time to come in here today, but I'd also like to thank all of our colleagues from the Department of Agriculture and from the wider community for making the time to come today. We think it's really important that we do celebrate what we're doing, but also it gives us an opportunity to talk about where we are at the moment, but where we could be in the future. We've got a large number of industry people within the room as well, and I'm not going to go through them. I've got a list, but I'm not going to go through them at the moment. But thank you all for coming in today. I'd hope today that you'll be able to take the opportunity to mix with the other colleagues in the room, to look at what the opportunities are, to look at what we've built here with the Department of Agriculture and Fisheries, and see that as a model for going forward in terms of collaboration. Because as we all know, collaboration is the future. We need to be able to collaborate really highly and very effectively with a whole range of industry, state governments, federal governments and universities, because we all have a part to play. The second broadacre cropping initiative maturing in the research strategic goals and delivering impact of research and support for our staff and our facilities. So enough of the past. What's the vision for the future? So I think this is an opportunity for us to turn around and look, where do we go from here? We need to continue to expand and broaden our opportunities for research collaboration, as I've already mentioned. The university and the institute that I represent aspire to diversify our research portfolio into other crops and other areas, and also build our capacity in areas such as protected cropping and also digital solutions for agriculture. So we're using this initiative as part of what we're trying to do there. We need to maintain and increase the impact of our research. As the Vice-Chancellor has mentioned before, the universities are under increasing scrutiny to make sure that their research is impactful. And so people are not want to just have research for research's sake. They want to see what the impact of that is. And the broadacre cropping agreement allows us to actually ground our research and to make sure that we are doing impactful research. And I really thank our colleagues for helping us in that journey from DAF. Gives us the opportunity to leverage opportunities. Leverage is also a very important part of what we're trying to do now through research. The funding for research has decreased from public purses, but now we have the opportunity to start to leverage what we're already doing, to work with numbers of partners. This also helps us to get greater impact and also to be able to do more. So we are excited to have opportunities to leverage with organisations such as GRDC, AgriFutures, but also groups like the Australian Research Council and the Future Drought Fund, that we have a hub here. So it's very important that we do regionally relevant research, which crosses right across the whole zone. It is extremely relevant to Queensland, but it's also relevant to other regions in Australia as well. And so we can be the conduit into changing practice across the whole region. So on a personal note, I'd like to thank our colleagues from the Department of Agriculture. It has been really interesting to see the maturity in this cropping initiative, how it's changed over time, how we're now working through the steering committee and the operational committee. The collegiate approach to this collaboration that we have witnessed from Department of Agriculture has been really refreshing and has been really a great experience for everybody that's been involved. So once again, I'd like to thank you for your attendance today and hand back to John.
Thank you, Gavin. And I think we saw one of the reasons why Gavin is the director of the Institute, with some great vision and leadership, not just of this agreement, but of a broader portfolio of activities in this area. So next, I'd like to introduce Dr. Chris Downs, who's the General Manager for Crop and Food Science in the Queensland Department of Agriculture and Fisheries. Chris.
John, thank you for your welcome, and thank you for this opportunity to be here today. I really do appreciate the opportunity that we're having to showcase the research that's taking place as part of the DAF USQ Broadacre Cropping Initiative, BACI. So as we look to the future, Australia's food and fibre industries will be shaped by multiple interacting changes that are coming at us over the decades ahead. Now, we know that empowered customers, consumers, global market dynamics, and the race to do more from less, together with climate change and disruptive technologies, will all impact the direction of the agriculture and food sector. These so-called megatrends, originally developed by CSIRO, updated recently by ABARES, will significantly impact broadacre cropping in Queensland. With income growth across the Asia-Pacific region, empowered consumers will have greater expectations around provenance, traceability, ethics, environment, and food quality. There will also be greater demand for protein from both animal and plant sources. The 7 most influential global giants, countries such as the US, EU, China, Brazil, India, Indonesia, and Russia, who represent together 2/3 of the world's population, represent large chunks of economic activity and food production as well. But however, with that consolidation, if you like, there is risk around volatility in global trade. And as a result, Australian companies are increasingly looking to onshore food ingredient development using Australian grown crops. Onshore Queensland's food ingredient development using Queensland grown crops. So there's the opportunity for us as we look up the value chain. There's also a growing focus on innovation and greater production efficiencies, new farm technologies, new varieties, greater yields, lower agricultural inputs, and less waste. They're all on the agenda, and together there is this constant demand for value and efficiency across our supply chains. So of course, as we know that climate is changing, as well as other changes in Earth systems that are quickly gaining momentum. And in terms of disruptive technologies, data, as Gavin has said, and information is radically changing the way that we breed new crops, we optimize our farming systems, the way we harvest our crops, the way we manufacture foods, and the way in which we engage with our consumers. And indeed, there are many that would say that the information component is becoming the most valuable ingredient in our food system. And how do we then factor that in and integrate that into the way that we work and the work that we do? So DAF has set our strategic direction for the next 5 years to meet a focus on consumer expectations, on building a more resilient sector, and developing innovative and globally competitive agribusinesses. Environmental and social goals are also vitally important. Now, in crop and food science, our work is concentrating on plant protein value chains and developing resilient cropping systems that are enabled by ag tech and data sciences. This is especially in this context of a variable and a changing climate. DAF's strategic relationship with the USQ and the investment that we make in this relationship in the Broadacre Cropping Initiative is playing a vital role for us as we explore areas such as crop protection, crop systems modelling, and investment into ag tech and data sciences. In the crop and food science area, we're keen to explore areas such as cropping systems and new genetics for heat and drought stress management, improving system responses to climate variability, understanding effective plant protection strategies in a variable climate, and in new cropping systems. We're interested in understanding the importance of new agricultural technologies, as I've said, and how the digital and data systems will support those cropping systems. We're interested to see how we could work together to diversify and develop new types of broadacre cropping systems, and ultimately also to address soil health and net zero emissions as well. So our important relationship with USQ and BACI positions us well together, I believe, very strongly for the future, positions us well for the development of resilient agriculture and food sectors for the future. This relationship in BACI is a vital investment with government, with academia, and with industry, so that together we can focus on these strategic priorities and we can support Queensland's regions and economy. So thank you. Looking forward today, looking forward to hearing about the research that will be presented, and I wish everyone well for their presentations. It's going to be a great day. So thank you. Thanks very much.
Thank you, Chris. And I think we've just heard that this research doesn't just cover the plants, it's the soil, it's the information, it's the climate, it's the technology. So this is actually something that is really significant in terms of the future development, not just of research in agriculture, but research right across the university. So thank you very much for that great insight. Everyone, that concludes the official proceedings for this morning. Thank you for joining us. I hope most of you will be able to stay for most of the day and see the presentations and enjoy the rest of the BACI Research Showcase. Thank you. No. There was another page which I hadn't realised. I have to hand over to the Director of the Centre for
Thank you, John. Thank you, John. Welcome, everyone. Welcome to the first of two sessions today that include lectures, hopefully short and sharp lectures, to introduce a number of BACI projects to show you what is happening on the ground within this partnership. We prepared 5 lectures for the first session, which is entitled Crop Health, not just because all the speakers come from USQ's Center for Crop Health, but of course also because they will talk about crop health issues that we don't want to have, but they do occur in the paddocks, and we would like to show you how we try to deal with these issues. So these presentations won't be data heavy, will be just snapshots, and it looks like we are well ahead of time, so there will be plenty of time for questions. So please ask questions after each presentation. Our first speaker is Dr. Dante Adorada, a research fellow from Center for Crop Health. Dante, the floor is yours.
Thank you, Levi. Good morning, everyone. Today I'll be presenting a new crop that must be not familiar with everyone, even me. The only time I got to know this crop is when I started working on it. So, sesame. Sesame is one of the oldest oilseed crops, widely adapted to subtropical and tropical regions around the world. It yields between 1,200 to 1,500 kilos per hectare in irrigated areas, and between 800 to 1,000 kilograms per hectare in rain-fed environment. So why are we looking at sesame? It's an important, we plant it because of their edible seeds and edible oil. So you often see them on top of the bread of your McDonald's burger. It's used for tahini, and it's used as a confectionery on candies and breads. And it also has recently been found to have ingredients in cosmetics. And it has antibacterial and antifungal effect on skin diseases. And it also has an excellent source of protein, fiber, and healthy fats, and important for immune system function. Currently, can you hear me? Currently, we are at the bottom of the pack of those importing in seed import globally. But since 1966, we are importing our sesame and sesame product requirements amounting to $20 million per year. Just recently, in 2020, we've imported 57.25 million sesame and sesame products. In terms of seed production, we aren't a member of those growing sesame nowadays. And most of the sesame production is dominated by African and Asian countries. Tanzania and Sudan are the two topmost African countries producing sesame and supplying the world. So what's happening? What's the current situation in Australian sesame industry? In 1990, there was an attempt to start on an industry, but it failed to establish simply because of poor genetics, and we don't have, we lack the facilities. Most of the countries now are doing harvesting sesame by hand. So there's no mechanically harvested sesame at present. In 2018, there was renewed interest on sesame here in Australia. The industry has re-emerged with the introduction of new varieties, which are mechanically harvested, high-yielding, and non-dehiscent or non-shattering. The opportunity for the sesame industry came up with, as influenced by the following factors. The climate here in Australia is suited for growing sesame. Sesame is drought tolerant and heat tolerant. It's suitable, we have suitable land and cropping systems here in Australia, and the facilities we've been using now and available now for summer crops can be used for sesame production. And also we are close to the Asian markets, China being one of the top Asian countries importing sesame. And also there's a market demand for quality produce. We often boast of Australian produce being the cleanest in the world. Where can we grow sesame in Australia? So initially, sesame was trialed in the Northern Territory and Northern Queensland, but the plan now is to extend planting sesame in Queensland all the way down to Northern New South Wales. So what's the potential of sesame here in Australia? Northwest Queensland alone is potentially worth 250 million based on some studies. In a BACI project that we've started in 2020 to 2021, I did some literature search and we found that globally, there are 30 diseases reported to be affecting sesame. We've done unprotected sesame crop plots here in southeastern Queensland, namely Alora, here in Toowoomba, Brigalow, Nandai, Jandaway, and Fort Martin. So the idea is we plant unprotected sesame plots, 30 square meter, just so to capture any disease that will affect sesame. And using, so we monitor those plots fortnightly on each season, and the diseases were identified morphologically and using molecular methods. And we found 12 fungal pathogens and 6 pathogenic bacterial species. So among those that we found, what could be the potential major diseases? We found Fusarium wilt, which was reported to be causing 45% yield loss in sesame. Alternaria leaf spot, that can cause 20 to 50% yield loss. Charcoal rot, which is very common in summer crop products, can cause between 5 to 100%. And Phytoplasma, that causes phyllodes or the witch's broom, can cause up to 80% yield loss. Interestingly, these diseases that we found here in southern Queensland were different from the one previously reported in literature. So as mentioned before, it was trialed in Northern Queensland and Northern Territory, and most of the diseases found in those areas were foliar diseases, simply because there's enough water to produce the disease. So what's next? Now that we know who our enemies are, we've lined up research looking into surveillance or what could be the other diseases. So we still need to do more surveillance. we're going to be testing different management methods such as chemicals, biological control, and disease resistance. All of these plans are outlined in the Australian Sesame Strategic Rdne Plan 2021 to 2026. This was developed in consultation with Sesame stakeholders group, and the proposal was submitted through the ASIDA, which is the Australian Sesame Industry Development Association, formed in March 2021. All of this has a goal of achieving a 10 million gross value product for Australian sesame industry so that we could be sustainable or even exporting sesame in the future. So I would like to acknowledge the support from Queensland, Department of Agriculture and Fisheries through the BACI initiative with the University of Southern Queensland. Thank you very much for your attention, and I'm ready for questions. Yes, I was just talking to Graham this morning, and they have trials in Georgetown looking into herbicide. And I know Tony Matchett is doing some herbicide resistance studies as well. And there's a person from Kwafi who just got in contact with me asking for seeds to do some work on herbicide resistance as well. So that's a priority. And all of those plans are outlined in the Rdne strategic plan. Yes, sir.
So how many hectares do we need to increase in GDP of $10 million?
Oh, okay. I don't have that figure with me right now. The Australian, the Rdne plan should have those information. I haven't gone through that one yet, but I think it's available now online. The contact person should be Olivia Reynolds.
Just a question. I was curious with some of those sites, because one of our concerns has been that, well, this is my personal opinion, I'm concerned about its adaptability, sesame, to the heavier soil types. I did see you had some heavier soil types there. So what's been your experience with establishment there, and the crop actually surviving rainfall events on those heavier soils? Were there any issues that occurred because of waterlogging, for example?
That's a good question. Okay, the one that we planted in Elora, really, it grew very well. The only problem we had on that one was we had a frost damage in April. So the crop was damaged and we didn't get to harvest it. We had problems in, what's the other one? Sorry, I forgot to. One of the sites, the problem was availability of soil moisture, and it didn't rain. That one was in Jandaway. So we had to bring water just for the establishment. Although the crop is drought tolerant and heat tolerant, as most crops, it always needs moisture and rain to establish. In Brigalow, it grew very well. Sorry, in Elora. In Brigalow, there's been some issues also. And I have there were recommendations by Ross Pomeroy, because he's the one who's really looking at it closely. And he's got recommendations about the soil type and the row spacing. So that should be in the BACI report that I've submitted just recently.
G'day, Dante. Paul McIntosh, everybody. Dante, certainly Sesame's been on the front of everyone's lips, trying to use that product more in our summer summer grains program. We know that Texas, they grow it fairly successful after a few years, and we know that Israel's pushing it the heck like heck. My concern is it's not so much the diseases, all crops have got a fair few diseases, but what about the shattering of those crops at the end and that grain loss right before you want to harvest it, the grain loss sort of stuff. And as someone mentioned, the establishment of that seed in the ground is pretty tricky sometimes too.
Could you say the question, the last one?
Question is, diseases are one thing and it's not your forte, what I'm talking about, but what about shattering of the plant just before harvest that can drop a lot of seed on the ground?
Okay, yeah, that's the reason why the crop failed in the 1990 because of the hissing or shattering sesame variety. We now have varieties or germplasm that we got from Acre. I think it's a US or Israeli breeding company, and they were saying that it's non-dehiscing. So the plan now is to plant this for adaptability study and check if it's really non-dehiscing, but that is not part of the plant pathology program. It's part of the whole program. So adaptability, agronomy, herbicide, plant pathology, the diseases. So it's all part of the whole strategic plan.
Hi, Dante. Is it time? The diseases you found were different to the north, but how different are they to other production in Africa or Asia? Is there any similarity?
The more tropical areas where there's supply of water or rain, that's where you find most foliar diseases and bacterial diseases. In areas where, like in Africa, where it's more on the drier areas, the problem is more on what we see here on the broad acre paddocks here grown for summer crops, which is drier. But those reported previously are just, they didn't really do any studies on that one. It's just merely detection of the disease, no further studies. So the one we're doing right now is looking into what's there and then continue the study. And we will extend that up in the Northern Queensland and Northern Territory.
Dante, with your isolation of the Fusarium wilt, was it Oxysporum or Solani or a combination?
It's, at the moment, it's Oxysporum, which is the more dominant species.
It was really nice to see this discussion after the lecture. And now we are move on. We have plenty of time. The official welcome at the beginning was 10 minutes shorter, so we can use this time wisely. Our second speaker is Dr. Nilufar Vagefi, Senior Research Fellow at the Center for Crop Health. Nilufar, over to you.
Thank you, Levi. Good morning, everyone. You hear me okay? Closer? Okay, is it okay now? Okay. So yeah, my name is Nilufar, and I'm going to talk about two different BACI projects that we have, which are focused on genomic research into bacterial diseases of Mungbin to support the Mungbin industry. Okay, so first a quick overview of the Mungbin industry. So Mungbin is the only summer pulse crop that is grown here in Australia, and the industry is mostly based in Queensland and northern New South Wales. Around 90% of the production is exported, returning on average of $1,100 per tonne to Queensland growers. So in 2020, the industry was valued at $118 million with a 5-year average production of 90,000 tonnes per year. So Mungbin itself, it grows quite quickly. From the time of sowing to harvest, it only takes around 100 days. So it's a quick cash crop, and it also has 2 sowing opportunities, both in spring and in summer. So it fits really well into the northern cropping system. And in addition to the high dollar value, Mungbin can also enhance productivity by fixing atmospheric nitrogen into the soil and therefore increasing the yield of the subsequent cereal crops. But Mungbin growers do face many challenges that threaten reliable production, because Mungbin crops can be affected by a number of diseases that affect yield. So here in these figures, you see two major bacterial diseases of Mungbin that can affect the foliage as well as the stem and can significantly reduce grain yield and quality. So these two diseases, halo blight and tan spot, can potentially result in 70% yield loss in severely affected paddocks. Both of these diseases are transferred by seed, so they are established in all Mungbin growing areas in Queensland. And unfortunately, once these diseases are seen in the paddocks, there aren't many management options available to the growers because there are no chemicals available for disease control. And another problem is that we don't have any completely resistant Mungbin varieties that's available to growers. So there are some Mungbin varieties that are less susceptible to bacterial diseases, but these are still affected. So there is a need to incorporate better resistance to Australian Mungbin varieties, because we know that varietal resistance can be one of the most sustainable and effective means of disease control. But the problem is, as I said, first of all, the resistance that is currently available is only partial. But another problem is that many of these Mungbin varieties, at the time that they were released, they were rated as moderately resistant to some of these diseases. For example, as you see on the table on your left, Solera and Crystal, there are two Mungbin varieties that they were rated as moderately resistant to tan spot in 2008. But then after a decade or so, both of these varieties are now rated as moderately susceptible to the same disease. So what is happening here? It seems like there is erosion of resistance in these varieties. And maybe the better question to ask is, why is this happening? So we think that there are potentially two reasons associated with this erosion of resistance in these varieties. First of all, this could be due to evolution of aggressiveness in pathogen populations, because we know that microbial populations, like any other population, they are unstable. They can evolve and adapt to control measures. For example, they can become more aggressive and then overcome crop resistance. But a second reason that could potentially have occurred here as well is that some of these aggressive bacterial isolates could have potentially existed in 2008. But potentially because of lack of knowledge on genetic and pathogenic diversity of the pathogen at the time, these more aggressive isolates may not have been incorporated into phenotyping efforts to breed for more resistant Mungbin varieties. So these varieties didn't really perform as well as they were expected to once they were commercially released. So we don't really know the answer to this question yet, but this is one of the fundamental issues that we're trying to address under these projects. So are pathogen populations evolving into more aggressive types? And the answer to this question is very important to be able to breed for durable resistance. So what we do here, we do whole genome sequencing of both historical and contemporary pathogen populations using our in-house sequencing facilities that we have here at USQ. And then we conduct evolutionary genomic studies to understand the adaptation of pathogen populations in Mungbin paddocks in the last few decades. We try to assess durability of resistance in Mungbin varieties by understanding how pathogen populations have been changing in the past and potentially evolving into more aggressive types, and also how they may change in future, to make sure that not only we breed for resistance, but we breed for durable resistance. Another outcome from these projects is improving integrated disease management strategies by better understanding pathogen biology and disease epidemiology, which can then be translated into reliable disease management recommendations. For example, if we know how or for how long these pathogens survive in Mungbin paddocks, or if we know on what other hosts they can live on or infect them, then we can come up with better informed management strategies for Queensland growers. So one of the most important aspects of these projects is to support DAF's Mungbin breeding to breed for resistance to bacterial diseases. And I think it's fair to say that currently the National Mungbin Improvement Program is DAF's flagship crop breeding program. And these two Mungbin projects that are funded by BACI are really good examples of how we can achieve more by collaboration between USQ and DAF, by sharing our knowledge and access to infrastructure, which will eventually achieve better outcomes for the growers. So, as I explained before, breeding programs can only be successful if they have a good understanding of genetic and pathogenic variability of pathogen populations. Otherwise, resistance will break down only a few years after the commercial release of the varieties. So under these projects, we work closely with DAF Mungbin breeders. We regularly update them from the outcome from our research, and also we provide them with the most aggressive bacterial isolates so that they can use them for breeding for resistance. Another outcome from these projects is delivering improved phenotyping methodologies. So we are working on molecular markers so that screening for resistance can be done in a more rapid and reliable way. So we are currently working on a disease assessment using a multiplex real-time qPCR assay. So Mungbin breeders can use this as a tool to screen for both bacterial diseases in the one assay. And of course, outreach and extension is an important part of our research here at USQ because our ultimate goal is to provide reliable information to Queensland growers and to the industry. So as a part of these projects, we will also be developing extension material, for example, fact sheets that we can then distribute to farmers at grower update events or at Mungbin research days to provide them with the results from our research and also let them know of the implications for disease management. And finally, thank you to our collaborators, especially Dr. Anthony Young from University of Queensland and Dr. Noel Knight from CCTM, Curtin University, and also Mungbin growers and agronomists for the support.
Within the bacterial population, we think there are hidden subpopulations.
Yes, I think, well, we find it mostly when we look at the historical changes. So we have populations from 1986, where first the industry, actually, when first these diseases were reported on Mungbin. And up to now, we can see that there were some subpopulations in the historical population that now dominate, and some of them are not on Mungbin anymore. In that sense, yet, but also when we look at the populations right now, there are a few different clonal lineages that we think they're slightly different in their aggressiveness as well. So yeah, we do find actually isolates that are the same pathogens, they're Curtibacterium, but they are not pathogenic on Mungbin and they're pathogenic on other crops. So there are differences in there too.
My turn. I said to Nilüfer when I broke in, what questions don't you want me to ask? Opal's not up there, Opal AU, because it's newness. Have you done any work on Opal AU with respect to halo blight?
Opal to halo blight? No, we have not done that yet. We have tested for transport because our halo blight project just started in November, really. But Opal is pretty susceptible to transport.
Sure is.
Yeah, but we haven't done the halo blight. I'll let you know as soon as we do that.
Thank you very much. That's extension for you, isn't it?
There are no more questions. We had a slide with that photo diagram. Yes.
Yeah, beautiful.
Can you explain why this slide?
Oh yeah, this is the right that promotes the growth of the plant. So they really grow very well under the pink light, which is basically just the wavelength that the plants use. So we have this, we call them CR, the controlled environment rooms at CCH, that really you can control the humidity and the lighting and the temperature very well. So at 27 degrees centigrade, they're very happy and nice and green, yeah. And then we can grow them under controlled conditions, and they really work well with inoculations too, yeah.
Thanks, Niloufar. I really enjoyed that. I guess I just wanted to let people know that I guess this season, for the first time, we're taking the methodology for that controlled environment room screen for TANspot, and we're sowing the first batch of those this week. So we're hoping to simulate what the results in the controlled environment rooms here, and basically embark on that journey of running breeding material both through the pot-based assay for TANspot and halo blight. We're embarking on that in March. So the results that Niloufar and her team are producing are pretty much going straight, being fed straight into the breeding program as soon as we can humanly achieve it. Thanks, Niloufar.
Thank you, Meryl. Sounds exciting.
Thank you once again.
Thank you.
Our next speaker is Dr. Rebecca Zwart, again from Center for Crop Health. Over to you, Rebecca.
Thanks, Louis. So good morning. I'm going to be talking to you about resistance to root lesion nematodes in chickpea. So root lesion nematodes are microscopic plant parasites found in the soil, and they feed on and reproduce inside the roots. So here in the photo, we can see a longitudinal view of a chickpea root. So the vascular tissue is stained pink in the centre, and on the sides you can see some nematodes and also some eggs. So the nematodes are migratory, and they move from the soil to the roots. The nematodes feed on the root cells, so they're causing damage to root function and prevent the plant from taking up adequate water and nutrients from the soil. And this ultimately leads to yield losses. So due to their wide host range, which includes many cereals and pulse crops, and widespread distribution in almost every cool, temperate and tropical environment, root lesion nematodes have a major economic impact on agricultural crops worldwide, including chickpea, which is an important crop to Queensland agriculture. So chickpeas is Queensland's most widely grown pulse crop, contributing 87% of all pulse crops grown in the state. Queensland is also the major chickpea growing state in Australia, with a 5-year production average of over 50% of the total Australian production. Australia is the second largest chickpea producer and leading exporter. So chickpea crops provide a substantial contribution to the Queensland economy. Furthermore, chickpea crops can fix atmospheric nitrogen, thereby improving soil fertility, which can boost the grain yield of subsequent crops, and thus contributes also economic benefits to the whole cropping systems. Chickpeas can be beneficial to cropping systems by acting as a break crop to cereal root diseases as well. However, when root lesion nematodes are present, the value of growing chickpea in rotation with crops such as wheat is dramatically diminished, because most chickpea crops are susceptible to root lesion nematodes and can suffer yield losses of up to 25%. Furthermore, high nematode populations build up in the soil and roots of susceptible varieties, and the nematodes remain in the soil to infect and cause yield losses to these subsequent crops. So root lesion nematodes are widespread throughout all grain growing regions in Australia, and surveys have reported about 80% of fields in the northern region are infested with root lesion nematodes. Growing resistant varieties is key to a practical, economic and effective control of these plant parasitic nematodes. The problem is that chickpea has limited genetic diversity due to domestication bottlenecks during the early origins of its cultivation. So the best performing chickpea variety grown in Queensland only has a rating of moderately resistant, moderately susceptible. So this means that breeders need to reintroduce traits into the chickpea gene pool in order for genetic improvement to be possible. So the Broadacre Cropping Initiative, together with GRDC and various national and international collaborators, as listed on the slide here, support a suite of complementary projects conducted by the USQ crop nematology team that are targeted at facilitating genetic improvement of chickpea for resistance to root lesion nematodes. So we are conducting the first systematic screenings of global chickpea germplasm to identify sources of resistance in wild chickpeas and landraces, and furthermore, genetically characterising the resistant sources by leveraging the extensive genomic data that has been generated on these collections by our collaborators. I'll give a little bit more information about these projects in the next slides. So also complementary to these studies are components of the next two projects listed, which have genetically characterised the best Australian commercial chickpea variety and the best advanced breeding line from the National Chickpea Breeding Program delivered through New South Wales Department of Primary Industries. So through these suite of projects, we are systematically covering all bases to achieve the best possible outcomes for genetic improvement. So as part of the national variety trials, the USQ crop nematology team provide national consensus ratings of varieties so that growers and advisors can select varieties best suited to their cropping systems. So ultimately, as the outputs of all these projects are fed into the chickpea breeding programs, the improved germplasm will filter back to us to evaluate through these national variety trials. So chickpea originated by selection from wild species growing in a very narrow geographic region in southeast Turkey, some 10,000 years ago. Up until recently, there were only 10 to 20 different genotypes of wild chickpea relatives actually held in global gene banks. So this situation has now changed with collection missions between 2013 and 2016, supported by GRDC involving Australian scientists from CSIRO and an international consortium led by the University of California, Davis, covering the remaining and diminishing biodiversity of these wild relatives. So seed collections from the wild chickpea plants have been processed through international gene banks and imported into Australia through the Australian Grains Gene Bank. USQ has been among several research organisations to first access this germplasm, and as it has become available through quarantine. So in total, we have phenotyped 329 chickpeas for the two species of root lesion nematodes that are prevalent in the Northern Grains region of Australia. So from the crop origin in southeast Turkey, chickpea has been taken to other countries where farmers have selected chickpea genotypes for a range of different environments. So in parallel to looking back to the wild relatives of chickpea to source novel resistance genes for genetic improvement, we're also evaluating a collection of landrace chickpea. So INCRISAT, the International Crops Research Institute for the Semi-Arid Tropics, have compiled a reference collection of chickpea that contains 300 genotypes from 29 countries, so indicated in orange on the map, and represents 78% of the genetic variability of the global chickpea collection of almost 3,000 genotypes. The advantage of identifying sources of resistance in landrace chickpeas is that these genotypes are the same species as a cultivated chickpea and therefore are favoured by breeders over wild relatives, as they may contain fewer undesirable wild traits. So the outcomes of our phenotyping experiments on these large collections is that we have been successful in identifying wild relatives and landrace chickpeas with higher levels of resistance to root lesion nematodes than currently available in our commercial chickpea varieties. Moreover, we have identified chickpea genotypes that have good level of resistance to the two species of nematodes prevalent in Queensland. We have also made considerable progress in the genetic characterisation of root lesion nematode resistance in the various sources, and we've been the first to report the genetic locations of mapped genes, as well as molecular markers and candidate genes for resistance to root lesion nematode in chickpea. So germplasm with higher levels of resistance than is currently available to breeders, together with linked molecular markers, will be valuable tools to improve the efficiency of chickpea breeding to develop and release root lesion nematode resistant varieties to growers. And also the identification of candidate resistance genes may also provide important insights for novel control strategies. So this work has led so far, as well as one article in press and more in preparation. So the root lesion nematode resistance ratings of the chickpea genotypes included in the national variety trials are directly available to growers through the MVT website. And also this research has provided opportunities for building research capacity and capability with one PhD student and 2 Masters of Science in Advanced Research. Okay, so thank you.
So what's the implications? Thank you. So what's the implications then in terms of nematodes in the whole farming system for being carried on chickpeas?
Yep. So any susceptible crop is going to increase the numbers of the nematodes in the soil. So if we can plant resistant chickpeas, obviously we're reducing the nematode reproduction. So we're getting fewer nematodes left in the soil. So then your subsequent crop, if you're planting wheat, then you've got, you're starting with a lower nematode population for any subsequent crops, which may also be susceptible to these nematodes because they have such a broad host range of many cereals and pulse crops. Really using crop rotation is an important control strategy, but there's limited crops and varieties that actually do carry those high levels of resistance.
So has there been any work done on subpopulations similar to the work that Nilafar was talking about?
Well, so far we're not seeing that there is a variation in these particular nematodes, across different states where parallel work, has been done.
So this was a very nice and comprehensive review of a number of projects, including BACI projects, because the BACI partnership has supported the work in nematology from day one, I guess, from 2014. Can you detail a little bit BACI's contribution in all these nematology projects?
So all of those projects that I had listed and the collaborators, so BACI is putting input into all of those projects, and particular, I guess, linkages with DAF in particular. We are using collaborating with the statistical support with SAGI for support of the statistical analysis of the trials.
There are no more questions. I don't need it. Thank you. Jan, thanks once again. It was a really nice presentation, and so many projects. You have seen so many logos there that all supported little bits and pieces. The next speaker is yours truly. And the slide is already there on the screen. So I will talk about a BACI project, a kind of small pilot project about fungicide resistance detection in Queensland broadacre crops. And I would like to start with highlighting something that almost everyone in this room knows very well. Fungicides are powerful tools, and sometimes the only tools that we have to control some of the most significant crop diseases. And year by year, when these chemicals are applied as sprays or seed treatment or in furrow applications, the target microscopic fungi that actually would cause disease are hopefully killed or at least heavily suppressed by these chemicals. So the disease doesn't develop in paddocks or is kept at acceptable levels. Now, over time, some subpopulations, Nilufar has introduced this term, so some subpopulations of the target microscopic crop pathogens have started to adapt to fungicide applications. And they survived the sprays. They do survive the spray every single season. And due to changes in their genomes, DNA level changes, they are able to become less sensitive to fungicides and finally completely resistant to some of the fungicides that we currently use in broadacre cropping. So this is how fungicide resistance develops, and this is very similar to the development of antibiotic resistance in those bacteria that cause human diseases and diseases of farm animals and so on and so on. So we know a lot about antibiotic resistance, and fungicide resistance is equally important when it comes to crop disease control. Here in Australia, we are very lucky because many years ago, a great team at the Centre for Crop Disease Management, Curtin University, Western Australia, largely supported by GRDC, have started a long-term project to dig deep into the DNA level mechanisms of fungicide resistance. And 2 years ago, they have started a national project, an extension and communication project called Australian Fungicide Resistance Extension Network, funded by GRDC. And we are part of this project. The most important outcome is that comprehensive guide that is on the screen, Fungicide. I can't read the title from here, but it's a comprehensive guide how to manage fungicide resistance if it has already appeared in some paddocks. So basically we have a problem that is called fungicide resistance, and we also have management tools to deal with this problem if needed. So via BACI project, what to do here within BACI? The issue is that basically we have no information about fungicide resistance in Queensland broadacre cropping systems. This might be surprising, but this map put together by Centre for Crop Disease Management, I think clearly shows the issue. All those colorful dots there show cases of fungicide resistance that have been detected and then confirmed in the laboratory with sophisticated molecular techniques. So as you can see, Queensland is almost like a blank spot. We only have there one single reported and confirmed case of resistance to just one fungicide product in barley powdery mildew populations. However, this is just an L, just a laboratory detection. It doesn't mean that the fungicides don't work in the field. Because of course, basically the direct outcome of fungicide resistance is failure, field failure. Fungicide applications simply don't work in the field. As anyone can imagine, there could be a number of reasons why a fungicide application fails. So it could be application error, it could be faulty product, weather, rain, wind, and so on and so on. So this is why we need laboratory background to clearly confirm whether or not a fungicide application failure is due to fungicide resistance in the field. And we haven't had this capacity here in Queensland. So the BACI project, which again is a pilot project with a modest budget, had the ambition and the goal to develop here at USQ in our molecular biology laboratories and other facilities, to develop and adapt methods that can help us detect the most common DNA-based markers that are associated with fungicide resistance in a number of broadacre crop diseases. And once these are developed, we would like to use them to monitor the so-called baseline sensitivity of some crop pathogenic populations to a number of important fungicides. The progress so far, so we have already adapted a number of methods, and we did check DNA samples from a number of crop pathogens coming from northern rice, and we will have a lecture about northern rice in the afternoon. I'm just flagging this to you. Also from barley paddocks and Mungbin paddocks. And I'm very happy to report that so far we haven't detected any DNA mutation that is associated with any kind of fungicide resistance in Queensland. But remember, this is a small project and working on a limited number of samples. However, while we were looking at this particular disease, which is there on the screen, which is called Mungbin powdery mildew, we had the feeling that we see field failures of fungicide applications. So the pathogen was happily sporulating and spreading after a number of sprays, which is a typical case of, or it could be a typical case of fungicide resistance, or it could be due to a number of other things, of course. So we sequenced the relevant DNA locus or the DNA region, and we didn't find any mutations there. Then a comprehensive search of the literature and also fungicide resistance markers in other crop pathogens, other powdery mildews that are closely related to this one, revealed that, oh yeah, fungicide resistance is very well known in this particular crop pathogen, but that particular DNA mutation has never ever been detected. So are we looking at the right mutation? That was the question. And in consultation with a number of industry partners and also friends at the Center for Crop Disease Management, the leader of that group, Dr. Fran Lopez-Ruiz, now we have the feeling that probably the diagnostics itself is not working. So probably we are not looking at the right spot in the DNA. So we have started to develop a linkage project proposal for the Australian Research Council with an important industry partner, because this industry partner want to have a clear and reliable diagnostic tool to detect fungicide resistance in this particular crop pathogen. So if this proposal is successful, should be submitted early next year. I'm looking at Maya, so maybe mid next year. So if this is successful, I think it will be a nice outcome of the BACI partnership, in addition to a few other outcomes. So these would be the outcomes of this project when it ends. Hopefully, we will have the laboratory background for the reliable detection of a number of DNA-based markers. So we need laboratory tools, and you have to choose wisely the workforce in the laboratory. You need someone skilled who can do the work for you over there. Of course, we want to even improve those diagnostic tools that are currently available in the literature. And overall, we want to improve fungicide use in Queensland agriculture. So in a nutshell, I tried not to mention any fungicide names in this presentation or any crop pathogen names, just a kind of high-level summary. This concludes this presentation. Thank you for your attention. Any questions, please do ask. And I'm also always looking at Kirsten, who is monitoring questions from our guests online. So if they're there, and yes, Paul, please.
Is there anyone else in this audience, just as a matter of interest? Thanks very much for that presto. I guess with my role in Pulse Australia, I'm aware of what's going on overseas, and some of the things that the drums are beating is about having our pesticide resistance be upon us, and also our MRLs, and that's more of a concern, the maximum residue levels. They're beating for lots of glyphosate and loxofop and a lot of other things, but they're also beating for chlorothalonil and mancozeb. So I don't know what we'd do if we didn't have those two in our armoury, because mancozeb's pretty handy with the multiple site of action sort of stuff. Have you got any thoughts about what the fungicide industry or the disease industry can do so we can protect our fungicides that we have and still make a marketable crop overseas when those countries, I said, close their doors to something like that?
Yeah, thank you for the question. This BACI project really doesn't focus on any issues like that. So yeah, mancozeb as an old product and a number of other old products, they will pose issues, and of course chemical companies will try to replace them. So our BACI project is really about detection of resistances towards the fungicides that are currently in use. That's what we are doing. It would be really important to extend this project and deal with mancozeb related issues and all the other issues, but I can't really comment on this from this BACI project perspective. Thank you. Any other, maybe someone wants to comment from the audience? Yes, Craig, please.
Yeah, Levy, just reflecting on your presentation, a number of presentations this morning. I'm just wondering whether there's areas that we're losing the battle against the various things that have been discussed, and what are those areas that we're losing the battle in, and what are the things that we need to do to maybe get on top of those things?
Well, the best resource we have at the moment is that guide, Fungicide Resistance Management Guide, that was published this year by GRDC as an output of the Australian Fungicide Resistance Extension Network. So there are a number of management issues there. First of all, we have to detect fungicide resistance. Once we know it's there, then the management issues are, of course, rotate chemistries, of course, use fungicides strategically, don't just spray on a calendar basis, and choose wisely your varieties, use resistant varieties if available, and so on and so on. Now, these management options should be applied even if there is no fungicide resistance. So that's a very important outcome of that guide and of the 2-year national project about fungicide resistance, that we should use fungicides according to those management rules, even if resistance hasn't been detected yet, to prolong, as you said, the lifetime of these chemistries, because we don't have too many at the moment. So is that a key area of exposure? Not really. So this pilot project is really about developing laboratory methods to detect DNA markers of fungicide resistance. That's all. Hopefully it will be extended. I'm very happy to hear that there's a lot of interest about that. Thank you.
There's a fair amount of fungicide being applied to broadacre crops. I mean, certainly in chickpeas as a standard, and in mung beans as a fair standard. Are you actually taking samples from paddocks that have been treated to test for fungicide resistance?
So we do collect samples, basically from paddocks where there is a suspicion that could be a failure of fungicide applications. However, this map actually has a number of chickpea diseases there. So fungicide resistance has been detected and confirmed in Ascochyta, for example, in chickpea. And just check the map. There are cases, none in Queensland, to my knowledge. Yes, Gavin?
So, Levy, as you get better at detecting fungicide resistance, do you think there will be more fungicide resistance out there?
As always. So that's why on this map, it's very important to note those 3 phases of detection. One is the fungicide works very well in the field. However, we do collect samples and we already see some mutations, which may already flag that the population has already included a number of pathogenic subpopulations which already have DNA markers that point to fungicide resistance. Second step is reduced sensitivity, which already the agronomist will notice in the field, hopefully. At the labor rate, the fungicide may not work properly, but at a higher dose, it still be effective. And then the third one is the complete resistance when the fungicides basically are useless, that particular fungicide product. So to answer your question, I expect some lab detections. I even expect some field failures, because I think there are field failures which are overlooked. And simply some fungicide products are inexpensive, so then another spray and another spray may still solve the issue. We don't know much about this. That's why I think this project is so important, to just start as a starting point in Queensland.
So it could be that it's similar to COVID. Nobody wants to get a vaccine until you've got it, but similar sort of thing with the fungicide resistance. We really need a fungicide resistant scare before people take it seriously.
Usually it is the case, yes. Yes, one case I think would highlight the importance of fungicide resistance in a number of broadacre crops. Yes, please.
Yeah, Graham Harris, Dav. I know, and it may just been oversight, I see peanut missing from there. Peanuts, have they been something you've been looking at? Because they have a heavy use of fungicide application historically.
So this map has all the dots, good as of July 2021. Mm-hmm. So this means that peanut pathogens may have not been sampled or may have not been, subject of fungicide resistance. Yeah, so this is an up-to-date map.
Yeah, we have a project starting in the near future in Northern Australia looking at dual-purpose peanuts. So that's with Central Queensland University. It may be an opportunity for us to keep an eye out there as well too, because that work will be done across Northern Australia with peanuts. So, and fungicides are heavily used in peanut production.
More than happy to receive samples. Thank you. If there are no other questions, then we will move to the last presentation today. The presenter is Dr. Cassie Percy, Research Fellow at the Centre for Crop Health. Cassie, over to you. You know how to use it. I don't.
Thank you, Levy. And good morning, and thank you for attending our presentations today. I would like to present to you our BACI project, which is entitled The Detection and Impact of Common Root Rot in Queensland Winter Cereals. Common root rot is a soil-borne fungal disease of winter cereals, and it's a visually difficult to detect disease in the field. Above ground, your crops may lack vigour, they could be pale in colour, have reduced tillering and characteristics like that. And however, it's below ground that where most of the damage is occurring. And the characteristic symptoms of common root rot can be seen in the image on the right, where you have extensive lesions on the subcrown internodes and root tissue of your cereals. These lesions can actually extend up into the crown region and up into the stem tissues of the cereals. And the symptoms are actually really similar to crown rot. And determining these diseases apart in the field is really challenging and requires pathogen isolation or DNA detection for accurate identification. Common root rot has been estimated to cause around 10% yield loss across Australia. However, plus or minus inoculated yield loss trials have never been conducted, and the relationship between resistance and yield loss has never been determined. During some national paddock surveys in 2018 and 2019, Stephen Simpfendorfer provided us with some data that indicated that 95% that the common root rot pathogen was actually detected in the roots of plants of 95% of the pathogens sampled in southeast Queensland. 67% of these samples also contained high levels of both the crown rot and the common root rot pathogen. Along with this, common root rot varietal resistance has declined. In 2015, at least 30% of our breadwheat varieties and all of our durum varieties had at least a moderate to high levels of resistance to common root rot pathogen. Now, all of our varieties currently are susceptible to moderately susceptible to common root rot. This image on the right is an image of a paddock of a badly infected common root rot wheat crop. This is what common root rot can look like. This is a susceptible variety, high inoculum levels and low moisture. This is not what we generally see, but this is what it can look like. So what is the impact of this increased incidence and reduced resistance to common root rot in Queensland? The first aim of our project is to determine what yield loss actually occurs in Queensland winter cereals. So common root rot yield loss trials are being conducted as part of this project across the 3 years from 2021 to 2023. In these common root rot yield loss trials, we have included all Queensland wheat and barley varieties that are available to growers as listed in the winter crop sowing guides, and the breeding companies have also been providing some germplasm that is soon to be released to Queensland. In these trials, we'll be looking at the disease severity, such as the lesioning on the subcrown internodes and stem tissues, and looking at how this relates to the actual yield loss in these varieties, and also looking at yield components. In 2021, we have 20 barley and 46 wheat varieties in plus or minus yield loss trials at Tasari Research Farm this year. Common root rot pathogen, I keep wanting to say its name, but we're trying not to do that today, was determined to vary in its ability to cause common root rot. And this was discovered as part of a experiment that was conducted in the under the GRDC National Soil-Borne Disease Interactions Project. So we tested 10 isolates from northern New South Wales and Queensland, and found that there was a significant variation in the ability of those isolates to cause common root rot. So the second aim of this project is to determine what the aggressiveness is of the isolates in Queensland. So we've been collecting isolates from Queensland. Stephen Simpfendorfer has provided quite a few isolates, and we're also speaking with the with the herbarium to obtain isolates that have been deposited there, and any isolates that come in from diagnostic samples will also be included. So at this stage, it's not what like Nilufer's been talking about, it's not a genomic study, we're just looking to see how much disease do these isolates occur. So we make an inoculum of each of those isolates and conduct aggressiveness testing in the glasshouse, and you can see in the image on the bottom there, this is an aggressive isolate compared to the control plant, and there's lesionings on the subcrown internodes and leaf sheath tissues, and overall reduction in size of the susceptible wheat variety there. So the isolates that are shown to be aggressive for common root rot are then used in all of our further glasshouse and field testing. So we also have a PhD candidate, Yi Zhong, who is looking at developing automated sensing techniques and machine learning algorithms to detect common root rot early in the infection. And Yi's been and will, she's been looking at taking measurements in the yield loss trials that I just described previously. So Yi is at present using the NIR near-infrared hyperspectral point-based sensor to collect data in both glasshouse and field trials, and also the Microsense dual multispectral camera to collect data also in the glasshouse and field, and this has been mounted on a tripod, as you can see in the image on the right, and also attached to a drone to collect data in field trials that way. So this project aims to evaluate the accuracy of detecting common root rot in field, to identify discriminatory spectral bands for common root rot, and also look at the timing of detection and whether these common root rot can be detected in this way early in the infection process. The final aim of this project is to then look at how common root rot and crown rot spectral signatures compare. So common crown rot spectral regions were identified as part of Jacob Humble's PhD here at USQ, and we would like to compare the spectral regions that are identified as part of these two projects to determine if the sensing techniques that are being developed here are disease specific, and can we differentiate these pathogens in the field. So in summary, this project aims to deliver a clear understanding of common root rot resistance and yield penalty in Queensland wheat and barley varieties, also to investigate the variation in the common root rot pathogen, develop sensing systems to detect common root rot, and determine if spectral signatures for common root rot are different from the visually similar crown rot. And this could be developed as a proof of concept tool for growers on farm. Thank you. Hello, Mandy.
I'll jump in first. Hi, Kathy. Richard Holstrank, GRDC.
Hey,
Kathy,
Richard.
my question to you, I guess, is ultimately on the yield loss component. Crown rot is, common root rot is obviously, the ultimate yield loss is going to be determined by environmental conditions at the back end. So how are you accounting for that, I guess, in your trolling methodology?
Yeah, sure, Richard. Like all of these fungal diseases, and we will get variation on the seasons. So yield data that we will be collecting will just be over the 3 years with the seasons that we get. But we do have opportunity to irrigate. This season's a lot wetter than potentially right now than I was hoping for, but we still, I think, will get good information across all seasons that are presented to us, and I think that's important too.
Cassie, thanks very much. It was a very nice presentation. I was very interested to see that the resistance wasn't really there in the current varieties compared to the older ones. Do you think that's because the breeding programs have stopped selecting for it, or has the pathogen changed?
I don't really know the answer to that, Mandy. I think it could, well, without a proper study, I don't think we can really say whether the pathogen's been changing, but it is not a focus of the breeding companies and hasn't been for a while. So there is a chance that that's starting to come through now.
Cassie, I'm interested by your detection technique, the camera system, and I'm interested in, once you can detect, how would you incorporate that into the management of the crop?
Yeah. I think because the pathogens are so difficult to detect, a lot of our growers out there don't really know what's in their paddocks, and they may, we can use predictive B to say that it's there or it's not, or it can also give us levels of vulnerability to particular pathogens. But I think this technology allows us to completely map paddocks. If we can detect where these infections are, and particularly if we can discriminate between diseases, then it'll be an amazing technique to be able to know what's in your paddock and then make management decisions around that knowledge. So.
So we still have time for questions and discussion. I'm just looking at Liz, but it looks like there's no question from our guests online. If anyone has any general questions or comments to the speakers in this session this morning, then now this is the best time to do it. Yes, we have already some interest, and in the meantime, I would really like to thank the audience for being so active and all these wonderful discussions after every single lecture. Yes, please.
A number of speakers have made reference to the pulse crops and leaving potentially nitrogen that's going to improve the yield of the following cereal crop. Now, in practice, I think the only crop we've found on the downs that's probably doing anything maybe faba beans. Chickpeas, mung beans, when we soil test afterwards, we are not finding nitrogen being left behind. Is there a gap in knowledge here, or is there should this be something we should be looking at? Because maybe I'll pass over.
There might be a response there.
Yeah,
Yes.
if I can just add to that, in our systems work, where you look at systems with more legumes versus less legumes, we're actually running it down with legumes, the nitrogen status, because people are growing legumes for money and they're not necessarily growing them when the nitrogen's low. So they're actually taking up the nitrogen and exporting more. And in the system, we're actually getting a counterintuitive result.
Okay. Any comments on this? I'm looking at Nikki without picking her.
I totally agree, Hugh, that yes, the biggest lever of nitrogen is faba beans because it's so related to biomass. So if we can get more biomass, we can get more nitrogen. That's really how the system works. But we need to be one, inoculating properly, checking that they're nodulating properly, and we need to be planting on low nitrogen, which Dave said. So all of those factors are where the legume comes to its fore in fixing nitrogen. It doesn't happen if there's nitrogen there, and it can use that. The whole reason that we can leave nitrogen behind is that it's not going to use stores of nitrogen, but if there's high levels of nitrogen, it's going to use that first. So they have to get that balance, and we're looking at that in mung beans in particular, but I think there are other crops that we could look at, new crops, potentially, to try to see what they leave behind in nitrogen as well. And so other candidates in the new scene like pigeon pea that we could potentially get a bit more nitrogen out of, that would be nice. That's right. It depends what your purpose is, but we want more protein in our grain as well. So we do want high nitrogen grain coming off our crops. But if we, like the work we're doing in mung beans is showing that, yeah, as you add more nitrogen, you're just flipping the whole system to use the nitrogen that's available for the mung bean. So it's about costs and inputs and all of that as well that you've got to balance up how much nitrogen you want to put there, but where are you putting it in the system, after what crops and in what situation. I think the whole farming system has to be looked at in that case.
Thank you for this discussion. If there are no other general comments or questions, then we can conclude our first session here. Before we wrap up, I would like to thank Liz, Kirsten, Janita, and Will, who helped us during this session, and hopefully they will stay with us in the afternoon. There will be lunch outside in the foyer where you had your morning tea, and from 1:30 we will continue with session 2 with 3 more exciting talks about BACI projects, and not just the particular projects, also about the framework within these BACI projects are operating, and then an exciting panel discussion about major questions for agriculture in Queensland until probably 2030. So please enjoy your lunch, and please be back at 1:30 when session 2 will start here in this room. Thank you, and enjoy your lunch.